I've spent the past 25 years studying the nervous systems of tiny worms, of an animal with 302 neurons. And I am struck over and over again by the complexity and versatility and flexibility of its tiny nervous system. And so I think that this problem is not going to get any simpler as we move up to more complex nervous systems. But I do think that studying this much more experimentally tractable and much easier to understand at a detailed level system poses for us the problems that we will have to think about as we try to reach the same levels of understanding of more complex brains. Right? So nowadays the number that people seem to throw around for the human brain is 86 billion neurons. I have no idea where that number comes from. But in any case, billions of neurons, trillions of connections. The animal that my lab studies, the nematode worm Caenorhabditis elegans, which, for those of you who don't know, is about 1 millimeter long, the size of a comma on a printed page, lives in compost, lives in gardens, lives in association with human agriculture, eating bacteria. Optically transparent, so you can look through it and see all of its internal organs when you look through a microscope. And the appeal for us as an animal for studying behavior is that its nervous system consists of exactly 302 neurons. And those neurons can be recognized in every single worm. I can look at any worm under the microscope, point at a particular neuron. Every neuron in the worm has its own unique 3-letter name that tells it from all the others. And those neurons are reliable in their position. They're reliable in the genes that they express from animal to animal. They're reliable, as I'll tell you, in the functions that they have from animal to animal. And importantly, in the connections they make with other neurons. And so in terms of having a cellular map and a structural map of the nervous system, this is by far the best understood of any animal. So at a level of comparison, when you get up to Drosophila, the fruit fly, you're talking about 200,000 neurons instead of 300. So it's a substantially simpler animal. Now, one of the things that is a surprise, came as a surprise to me and even to other people who love the worm, is that the genome of this animal is actually not that simple. That despite its incredibly stripped-down nervous system, it has almost the same number of genes as a mammal. It has 20,000 genes instead of 25,000. And even more sobering, this includes almost all of the genes that you might actually care about if you were studying the nervous system, with a couple of notable exceptions, including, for example, almost all of the genes that have been implicated in human neurological and psychological and psychiatric disorders. So the building blocks have not changed that much. The core components that are used to build brains have not really changed that much in the past 800 million years of evolution. It's the assembly of the structure into more and more complex structures. I think the simplest hypothesis for where the much greater capability of complex brains come from is really that it's the larger number of neurons and the larger number of connections that give rise to more sophisticated properties, not the components themselves, not the individual components. So here is an illustration. The slide shows a particularly pretty diagram of the C. elegans wiring diagram, a map of the cells within the worm's brain and its synaptic connections. Every single cell is shown here. This structure here is meant to show you a couple of things. You're not really meant to be able to read the names of the cells. What you can see is that there's some that are red. Those are sensory neurons, and they're about a third of the neurons in the worm. And then there's a set that are green, and those are motor neurons, and those are about a third of the neurons in the worm. And then there's a set that are blue. And those blue ones are neither sensory neurons nor motor neurons. They're the ones in the middle that are involved in collecting information and transferring information between different sets of neurons, presumably the sensory motor transformations. Now, the light gray lines, which you may or may not be able to see, are the connections between them. And what you should get out of this is that the diagram here indicates something about the relative distance between neurons. And the order tells you that the information is mostly flowing from sensory to motor. But the very large number of gray lines tells you that these neurons are very densely connected. There's not like simple through pathways from A to B to C. There's a lot more networking among these neurons. There's incredibly heavy connections between them. And we think that's probably important for their functions in ways that I think people have not appreciated in thinking about this nervous system. So when you study a nervous system like C. elegans, you can make use of the animal's behavior to ask what neurons do and how they're assembled together. And all the behaviors I'm going to tell you about today involve the worm's responses to smells in its environment. So it lives in the soil, it's looking for bacteria. Odors are its richest source of information about the world. It is more sensitive to odors that we have. To give you an order of magnitude, your nose has about 400 different genes for detecting odors, for detecting different chemicals in the environment. The worm has about 2,000 genes for solving the same problems. The worm is devoting 10% of its genome to figuring out what odors are out there in the world. It's amazing. And it can report what it smells to us by what it does. So, for example, some odors it finds very attractive, put 100 worms down on a plate with the odor on one side, and they'll all go there. Other odors it will find repulsive and go to the opposite side. We can also tell if it seems not to care, although, of course, if it doesn't respond, we don't know if it sensed it or if it just didn't have a preference. But this gives us a simple way of looking. We can look either at individual animals or at groups of animals to figure out how these behaviors work. And the first level of understanding these behaviors, again, is very orderly, which is that you can map the sensory neurons. I told you about a third of the neurons are sensory, and about a third of those are apparently olfactory and taste neurons that sense chemicals in the environment. And for every single one of them, you can identify a set of chemicals that it detects and a set of behaviors for which it is a sensor. So certain chemicals, for example, will be detected by the neuron in blue that's labeled AWA. On the left there, other chemicals will be detected by each of these other neurons in this cluster in the head, all of which have endings that poke out at the tip of the nose to detect odors in the environment. And their different patterns of gene expression allow them to detect different odors. And furthermore, in a set of experiments, you can show that they have a very orderly relationship to behavior. So the neurons that detect attractants are different from the neurons that detect repellents. And you can do fun experiments with these neurons where you can switch the animal's behavior by switching the receptor for a particular odor from a neuron that senses attractants to a neuron that senses repellents through a set of genetic manipulations. And you can then make the worms avoid the odor that they would normally be attracted to. So that very orderly map is really a map of molecules that detect the odors, the receptors, sensory neurons that then attach those odors to a particular behavior. A very neat representation of innate odor preferences in an innate map. And this idea of innate behaviors and an innate map applies at least to some extent to the nervous systems of mammals as well. And the clearest example of that is the taste system. Mammals are born knowing that some compounds are toxic and should be rejected, and other compounds are nutritious and should be accepted. This is done in part through cells in the taste bud in the mouth and in part through olfactory neurons. And these taste cells for sensing nutritious compounds like sugars or amino acids are different from the taste cells that sense toxins or alkaloids, which are sensed as bitter. And really nice work from Charles Zucker and McGreeba's labs have shown that there too, the identity of the cell, the taste cell, is directly wired up to the innate acceptance or rejection behavior. That if you switch around receptors between these cells, you can make mice drink strychnine for a while. And you can make them reject sweet substances, or you can make them taste or respond to completely unnatural substances that no mouse has ever seen in the history of the world. This is a very kind of pleasing, orderly view, again, of the way that chemicals are sensed. And at a philosophical level, it really gives the idea of some sort of a fixed pathway for innate behaviors. And I include here this image from René Descartes, who really first expressed this idea that all information reaches the brain through the senses that represent information about the world to us. So while these are real results and these are real experiments that you can do, these are constructs that we have imposed on behavior. That when we study behaviors in the laboratory, we go to an enormous amount of trouble to make them very reliable and to make every animal behave exactly the same. And by doing that, we tend to find that we can find very reliable pathways to carry them out. But I think that we then conclude that that's the way that behavior works. But in fact, we're ignoring an incredible amount of complexity of behavior, even in the simplest animals, and an incredible amount of variation in behavior. And that's the main point of my talk today, is to point out that variability. And to point out that behavior is not really so much Cartesian as it is sort of a middle-aged Walpurgis knot of activity. That, on any given night here on Mardi Gras, actually, there will be all kinds of people eating and drinking and carrying on, showing all kinds of different behaviors, some of them relevant to the context, some of them relevant to internal drives. Obviously, the next day, Lent is going to start, everyone's going to go to church, their behavior is going to be totally different. And it's really accepting this kind of wild variability of behavior that is going to be necessary for understanding what the brain is doing and for generating meaningful maps of the brain. So, and what I'm going to tell you is that this is true even for a worm with 302 neurons, and that we understand a little bit about how those different kinds of variability are often quite similar to those of more complex animals. So, first of all, it's important to recognize ways in which behaviors can vary systematically. So in C. elegans, we know of 3 different ways that we can change the olfactory preferences of animals that are outlined on this slide here. The first is that there are changes that occur in development that can change the behaviors and even, in fact, the morphology of the nervous system of older animals. So very early larval stresses cause animals to take alternative developmental pathways. There are neurons that look completely different depending on whether those pathways are carried out. And even if the animals recover from that stress and become adults, their behaviors, their patterns of gene expression are different from those that they would have been otherwise. So any map of the brain is going to have to take into account the differences that occur in different individuals because of the developmental changes. The second is learning. Learning is a fundamental feature of the nervous system. The nervous system's job is to predict the future. It does that partly through information stored in the genome, like sugar is good for you and strychnine is bad for you. And it does that partly through matching your own experiences to what's occurring in the future. And even the simplest animals, like worms, can learn important things about their environment. So worms will learn to approach odors that have been paired with food, ignore odors that have been seen in the absence of food, and actively avoid odors associated with food that's made them sick. So the preference for odors is modified by an animal's experience, and this is likely to also change the map of behavior, even in the worm brain. And finally, behaviors differ between individuals. And when we look at wild-caught worms from different places around the world, we find that these wild animals behave differently from one another when we move them into a laboratory. The natural variation between them causes them to have different kinds of odor preferences. I've shown a drawing here of a behavior that I'll get back to later. It's an illustration of the animal's preference for other animals, whether the animal wants to aggregate or be in isolation. These kinds of variations are systematic in all animals, and they're well recognized as being systematic in human brains and other animal brains. So any map that we get of the nervous system is going to represent one out of a set of maps. For each of these 3, you can say that there might be some sort of structural manifestation. Development can change the structure of the brain, learning can change the structure of synapses, genetic variation changes the structure of genomes. And so to some extent you can say, well, this is a sheet, which is to say that, well, there'll be, once you understand the structure of the brain, then you can completely understand its function. But I want to say that that's not true either. And again, that's not true even in the simple nervous system of a worm. And that's represented in exactly this sort of stepping away from the idea of innate, reproducible, perfectly reliable behaviors, and looking instead on what animals do in more ambiguous circumstances. And so we can set up experiments where we take animals that are genetically identical to each other and then expose them to the same stimulus over and over and over again and ask them what they do. And this table here illustrates a particular odor stimulus and the animal's response to that odor stimulus. 23 individual animals, each one row of the table, were exposed to the same odor stimulus 24 times. And over the entire population, 30% of the time, the animals moved forward, 25% of the time, the animals reversed, and 45% of the time, the animals did a complicated reorientation maneuver called a pirouette. So if you look at these numbers, they're reliable across the population, and they're also characteristic of each individual animal. Each animal responds to that odor a third of the time by moving forward, and 45% of the time by pirouetting. We don't see any statistically significant difference between these genetically and environmentally identical animals. They are, on a trial-to-trial basis, responding to the odor or not responding. And we also don't see a systematic change over time. The 24th stimulus can be the one that stimulates the animal. The first stimulus can be the one that stimulates the animal. So there's something almost like a lottery going on here for the behavior to take place. So one of the questions we've been asking is where in the circuit we can find that kind of variability. So here we have this wonderful wiring diagram of the worm. We can take the sensory neurons, which we know what they are. We can take the motor circuits, which we know what they are. There's a neuron called the backward command neuron, for example, that's responsible for the reversal behavior, which is one of the options that the animal had. And we can ask, using sort of an activity map kind of experiment, where we look at when these neurons are active and on which trials the animals had neural activity, and ask how that correlates with the animal's behavioral outcome. So this next movie isn't going to show, but what we can do is we can express genetically, under controlled conditions, a reporter of neural activity in each of the different neurons along this pathway. We can allow the animal to move freely around the environment, expose it to these odors, watch what it does in terms of its behavior, and then watch what each of these neurons along the pathway do. And try to relate neural activity to the animal's behavioral outcome. And so if we start, for example, with the olfactory neuron, we find that the olfactory neurons respond to the odor every single time the animal encounters it. So the variability is not something about the neuron becoming sensitive or insensitive. Every worm at some level knows that that odor was there because it reports that in the activity of a particular neuron. This particular table is actually showing you a single animal tested 80 times with that same odor pulses. So that in a single animal, we can get the kind of statistics where we can ask what did that animal do and what did the neuron do in each of the different circumstances. And so we can separate out all the trials and say, well, there were 30% pirouettes, 50% reversals, 20% forward movements, same general statistics. Every time the sensory neuron was active, and furthermore, when we average it, those three curves are indistinguishable. They're completely superimposable. There's no difference in strength of the sensory input that can explain the behavior of this animal. Yeah?
What would they do if there wasn't a odor? What would the default behavior?
The default behavior would be they would be performing pirouettes and reversals, but they would not be time-locked to the stimulus. They would be performing these behaviors actually spontaneously as they moved around the environment, on average about once a minute. And so it's really the time locking that tells you that this is an odor-evoked behavior. And that's actually probably directly relevant to the answer to this question, right? So we can look through each of the neurons that way systematically. And what we find is that the sensory neuron responds to odors every single time. And if we go down to the command neuron, it's active at every single reversal. When we go to the intermediate levels, what we see is the variability being expressed through different intermediate levels. And what they're doing is it's not that they do or don't respond to the sensory neurons. It's that they respond to the rest of the state of the animal at a behavioral level. That the state of the larger circuit and the activity of other neurons can be used to predict whether the downstream neurons are going to respond to sensory input. So there's a certain, I've illustrated that as indicating feedback from the command neuron to this intermediate neuron. It's actually something a little more complicated. It's really that whole network of gray lines that were in the earlier slide that I showed you. But that information from many neurons is affecting the flow of information through any one neuron. So the activity maps are changing dynamically. Activity is not going to flow through the nervous system reliably in a way that you can look at just from the connections. You're going to have to know the instantaneous state of activity to know where activity is going to go. So in addition to this sort of second-to-second way that the state of activity in the circuit affects the flow of information, there are also slightly longer-lasting ways in which information flow is changed. And these are context-dependent behaviors. So the behavior that I want to talk about is aggregation behavior. Social behavior is a feature of pretty much all animals and even most unicellular organisms. They can usually recognize other members of their species and either associate with them or not, or perform various kinds of coordinated activities as part of the group. So C. elegans is no exception. It has sexual behaviors, and it also has a very broad variety of pheromones that regulate its development and behavior and even its lifespan. And it also has a regulated aggregation behavior, in which animals can either choose to basically scatter from each other or to gather in aggregates. And this is a behavior that's affected by many factors, but within one animal, it can be rapidly regulated by stress levels. So when the animals are exposed to certain kinds of environmental stress, the one that we use actually is an oxygen stress that they don't like. They form aggregates. When the stress is relieved, they immediately disperse. And we can switch them back and forth between these behaviors within a minute, just by changing the oxygen level. So how do they do this behavior? Their response to one another is mediated by pheromones. So they detect pheromones that the other ones secrete. The animals within an aggregate are attracted to pheromones, and you can show in a chemotaxis assay that they will go to the pheromones. Animals that are not in an aggregate are horrified by pheromones, and this illustrates what they do if you put a pheromone in their path. It's quite clear that they are not solitary because they don't sense each other. They are actually solitary because they actively avoid one another, as shown here for a purified pheromone that each animal will be releasing constitutively. So here we have an animal that can switch its behavior from attraction to repulsion very rapidly on a minute-to-minute basis. So the question is, okay, now let's follow this map through. How do you get these two different behaviors? And the first answer is, whether we look at the attraction behavior or the avoidance behavior, it's always the same sensory neuron that detects the odor. So this is not a neuron for attraction or repulsion. This is a neuron for attraction and repulsion. It's the same neuron, regardless of the context of the behavior. So now we have to move from that neuron down into the circuit. And to tell you how we think this behavior works, I'm just going to mention a few things about the way the circuit works. Neurons have three basic ways of communicating with each other. The one that is studied the most, the one that is accessible to electrophysiology, is fast chemical transmission, which is the release of molecules like glutamate or acetylcholine that lead to rapid depolarization or hyperpolarization of neurons, and the transmission of information really at millisecond levels throughout the brain. There are also electrical synapses, gap junctions, that allow communication directly between neurons. These are relatively rare in the nervous systems of both C. elegans and mammals. They represent about 10% of the total. And then superimposed on both of these are neuropeptides and neuromodulators. The neuromodulators, serotonin, dopamine, norepinephrine. Neuropeptides, there are about 100, actually, in both worms and mammals. And these are small molecules that act not directly at synapses, so they can act at a distance, although they usually act reasonably locally. They act over a time scale of seconds or minutes because they act on G-protein coupled receptors and on the biochemistry of the cell typically, rather than directly depolarizing or hyperpolarizing it. And they tend to set the state of circuits in higher or lower levels of activity, or the strength of synapses at higher or lower levels of activity. Now, these have not been the most popular molecules for electrophysiology, and they're not accessible to anatomy, but they're extremely important in behavior. They're implicated in pain, in bonding behavior, sleep, hunger, stress, and emotion. So, of substantial interest. And when we look at the wiring downstream of the pheromone sensing system of C. elegans, and we follow it out, we find that different classes of synapses among that subset I just showed you are associated with the different behaviors. So there are actually different synapses required for repulsion and attraction. But there are a set of chemical synapses that ultimately end up with those command neurons that drive repulsion behavior, the backward command neurons. And that the same neuron also has a set of gap junctions attached to a different set of neurons that are required for the attraction behavior. So all of these synapses are present in any animal. The context is switching the animal between using one set of synapses and using the other set of synapses. And I just want to mention that the only way we can figure this out is by perturbing the synapses. We actually have to go in and shut different things off genetically or chemically to really separate out these activities. So we need a functional map, not just a structural map, not even just an activity map. And the way that the decision between these is made is by the third class of transmitter, that the role of the neuropeptide is to significantly weaken the gap junction circuit under low stress conditions. When low stress comes in, the peptide comes in, and one set of synapses is favored over another. And so the sets of connections that are active are being regulated on a minute-to-minute level by this other information that is flowing through in parallel to the second-to-second flow of fast electrical communication and sensory information from the pheromones. So what this says is that any nervous system, certainly the C. elegans nervous system, but I would like to argue all nervous systems, consists not just of one map, but of many, many maps. And that one of the ways in which these maps change is by rewiring circuits through the use of these neuromodulators. And just to point out that this is not just a feature of the very compact nervous system, it's clear that these modulators are important in feeding circuits. They're very important regulators of hunger and satiety in all animals. In sensory circuits, they're the key element that switches your light between dim light and bright light vision, are the neuromodulators dopamine switching circuits. In motor circuits, and importantly, it's worth remembering that drugs of abuse and drugs for treating psychiatric disorders are almost universally acting on these modulatory systems. Opioids, serotonin, dopamine, norepinephrine, and so on. So just to sort of summarize with an intuitive example, whenever you move through the environment, you're encountering sensory cues. Your brain processes that information differently depending on context. This is my lab barbecue from last summer. The smell of cooking meat, if you are really hungry, is highly arousing, gives a physiological response, also a lot of interest and attraction. If you're not hungry, those responses are substantially weaker. As I did last summer, you have norovirus, that odor elicits a very strong and immediate nausea response, right? Which wasn't there before, actually, right? So the same odor stimulus can be either highly attractive, highly aversive, or neutral, depending on the context and the way that information is flowing through the nervous system. I don't mean to say that the worm nervous system is all there's going to be in the mammalian nervous system. I am actually 100% sure there's going to be a lot more. But I don't think that all of this information was thrown out as the more complex nervous system was generated, any more than the basic machinery for things like DNA replication and the cell cycle were thrown out as animals became more complex. And more DNA had to be regulated, and the cell cycle had to be regulated in more detail. So I just want to close by saying I think we have to think about a lot of different maps of the brain. This is really the way that I think about the NIH Brain Initiative is that in order to understand the brain, we're going to need a lot of maps. We're going to need to know what the cells are, what the molecular components of those cells are, both because they determine the properties of circuits and because they're going to provide, in many cases, our understandings from disease mechanisms, our molecular understanding. We're going to need to know how these cells are connected to one another locally and at long range and how that changes with development and learning. We're going to need activity maps, electrical activity maps, but also chemical activity maps. I think we need to know how these modulators work and where they are and how long they're around, and that's not so easy to do right now. And multiple time scales and spatial scales. We need functional maps. We need to perturb this activity to understand how it's being expressed. And just a point that I think is very important is that activity doesn't tell you what's important. Information is broadcast across the brain. I would argue that every part of the cortex knows important things that are going on everywhere in the brain. The World Cup is coming up. Lots of people are going to be talking about it, but that doesn't mean they're going to be affecting the outcome. And in the same way, many neurons are going to be active in ways that are not directly related to whatever activity they're reporting. And finally, the real point is that we need conceptual maps. We're going to need theory and we're going to need understanding. that brain mapping is not an engineering problem, it's a scientific problem. It's going to take all of our creativity and insight to understand what the maps of the brain are to make progress. Thank you.